MicroRNAs are small RNAs that regulate the transcription of messenger RNAs. They play an important role in many signaling pathways, including those necessary for development. MicroRNAs have a seed region of 8 nucleotides that binds to the 3' untranslated region of a target mRNA. A single microRNA can have several different targets, and a single mRNA can be targeted by several microRNAs. MicroRNAs are cleaved from pre-microRNAs by an enzyme called dicer. Pre-microRNAs are cleaved from long primary microRNAs by the Drosha complex, which includes an enzyme called DGCR8. The role of microRNAs in early development has been examined through inducing both over- and underexpression of microRNAs in embryonic stem cells. Global underexpression can be induced by knocking out either dicer or DGCR8. A vector can be used to induce overexpression of specific microRNAs, allowing researchers to also eliminate the possibility that the observed changes are linked to a previously unidentified function of dicer or DGCR8. By re-adding microRNAs, researchers were able to infer which microRNAs were needed to restore wild-type growth. By analyzing the transcriptome, researchers were able to infer which genes the microRNAs regulate. The role in embryonic stem cells deficient in dicer, OCT4 levels used as a marker for pluripotency do not decrease as they do in wild-type embryonic stem cells, which will spontaneously differentiate over time in the absence of factors promoting continued pluripotency. Transfection with miRNA 290 cluster microRNAs leads to OCT4 levels dropping, although not back to wild-type levels. Similarly, DGCR8 knockout cells show inhibition of markers tied to differentiation and continued expression of pluripotency markers, including OCT4 and NANOG, as seen on this slide. Once again, reintroducing microRNA 290 to 295 is able to restore some degree of wild-type phenotype. Cells overexpressing microRNA 290 to 295 cluster microRNAs also show a delay in differentiation. The overexpression of the microRNAs may enable the cells to continue to divide more rapidly, even in unfavorable conditions. The dicer-deficient cells also do not proliferate as rapidly as their wild-type counterparts. Transfection with microRNA 290 cluster microRNAs restored normal growth. DGCR8 knockouts also proliferated more slowly and, compared to the wild-type, were more likely to be found in the G1 stage of the cell cycle. Addition of certain microRNAs, including 4 in the microRNA 290 to 295 cluster, was able to promote proliferation in both knockouts. 3 restored the proportion of embryonic stem cells in the G1 phase to near wild-type levels. Adding the microRNAs to wild-type cells had no effect. Cells with overexpression of the microRNA 290 to 295 cluster proliferated more rapidly than the wild-type cells at low nutrient concentrations, but replicated at the same rate as wild-type cells when nutrition was plentiful. At low nutrient concentrations, the microRNA-enriched cells were more likely to be in the S phase and less likely to be in the G1 phase compared to the control, the opposite of the knockouts. A short G1 phase is characteristic of embryonic stem cells, and it is likely that the microRNAs play a role in regulating the G1 to S transition. The effects of the microRNAs on cell cycle transition may be tied, at least in part, to P21, a protein that inhibits the transition from G1 to S phase and is inhibited by 290 cluster microRNAs. It has at least 2 target sequences, and its expression has been measured to be downregulated by these microRNAs. P21 blocks the formation of CDK cyclin complexes needed for cell cycle progression from G1 to S. Compared to somatic cells, embryonic stem cells divide rapidly, perhaps thanks in part to lower expressions of P21. It is also characteristic of P21 to induce cell cycle arrest at low nutrient concentrations, which was observed in the wild-type embryonic stem cells, but was not observed in the stem cells where the microRNAs were overexpressed, suggesting that these microRNAs may be inhibiting the expression of P21.